just to start off with, we talked briefly about pesticides in general. And pesticides are a broad class of compound that would include anything designed to control a pest, which could be an insect, it could be a weed, it could be a rodent, it could be lots of different things. But pesticides are something that there have probably been efforts to produce for as long as there have been people. None of us really want to be bothered by pests. So we know that, as early as 1000 BC, there's evidence that people were using sulfur specifically as a pesticide in China. By 1500, people had discovered that arsenic was really good at killing things, and they were using various different arsenicals as pesticides. By 1700, Europeans had discovered the New World and had found tobacco, and tobacco makes nicotine, which is a really good insecticide. So tobacco extract was being fairly widely used in some cases as a pesticide. By 1850, we had maybe the first green revolution. So there were a number of natural pesticides being used, including rotenone, which comes from a type of chrysanthemum, rotenone and chrysanthemum extract, which would contain pyrethroids. By 1900, a variety of arsenates were in common use, and some of them still continue to be used up until this time, although just recently the EPA has revoked the license for some of the weed killers that contain arsenic. In 1930s, DDT was first synthesized and used as a pesticide, as well as a number of other synthetic chemicals, including 2,4-D as a herbicide and the dithiocarbamate fungicides. By 1944, the chemists had gotten busy and made the first organophosphate, which was parathione. And then by the 1960s, we had pretty widespread use of carbamate insecticides, as well as the synthetic pyrethroids, so derivatives of the chemicals that were first found in the chrysanthemum extract. So there have been pesticides around for a long time, and as I said, there are lots of different types of pesticides. Mostly I want to focus on insecticides today, so we'll primarily talk about insecticides. But there are, widespread use of herbicides. We'll talk briefly about atrazine, which is probably one of the most widely used herbicides. Fungicides, avicides, molluscicides, and rodenticides. So lots of different pesticides. But we're going to focus on insecticides and, as I said, atrazine, which is an herbicide. So kind of like metals, pesticides have a lot of use. We know that there are billions of pounds of active ingredient made in the United States each year. Well over 300 different pesticides are registered for use in the United States. One of the things that's important to note is that over 50% of the use is actually non-commercial. And what this means is that basically as homeowners, it's you and I that account for over half of the use of pesticides. So agriculture and commercial use for termite control and things like that are big uses. But when you think about 300 million people, actually non-commercial use accounts for over half of registered pesticide use in the U.S. And we typically think about pesticides when we're talking about toxicity, in a bad way, but in an effort to be balanced, I think it is important to remember that pesticides do have a lot of benefits. So, you get higher crop yields, better health, right? So malaria and West Nile virus are pretty common problems that can be controlled with pesticides, or at least partially controlled with pesticides. And people recognize this, especially back in the 40s. And when DDT was first invented, people thought it was really a miracle in terms of its what it could do. And the Nobel Prize was actually awarded for the synthesis of DDT in 1948. So we're going to talk about DDT in a bad way, but remember that for a long time people thought DDT was the best thing since sliced bread, which is part of the reason we have the problem that we have today. All right, so if we think about insecticides, we know that insects are arthropods in the class Insecta, and the thing to remember is that lots and lots of arthropods share pretty much the same neurochemical and metabolic pathways. So insects would be the target organism in this case, but you have an awful lot of pesticides that are designed to work on insects, which are a type of arthropod, that also affect non-target arthropods. So things like crustaceans are also very, very susceptible to toxicity. So you see a lot of non-target toxicity with use of pesticides because basically they're the same type of organism. So even though a lot of pesticides we have today are designed to act specifically on the insect or the arthropod version of a receptor, they're non-target organisms that have very similar receptors, and we'll talk about fipronil as a really good example of this. One of the things to remember is that a lot of these arthropods, these aquatic arthropods, are not very visible. A lot of them are sort of small sediment-dwelling organisms, so people don't get that worked up about them like you would maybe with a fish kill. They're not visible. You know, you kill off all the amphipods. You don't see it initially. But a lot of these are really important in aquatic ecosystems. So they often form the base of a lot of the carbon cycle and perform a lot of activities like bioturbation of sediment. So turning that sediment over, making sure that process goes on, and helping to transfer energy, you know, from the production and sediment level back up into higher trophic levels. So non-target toxicity of insecticides can be a really significant ecological problem. So I want to talk briefly about a couple different classes of insecticide today. We'll touch on organochlorines, which are things like DDT, toxaphene, dieldrin, lindane, things that were really widely used in the 40s and 50s and pretty much up into the late 60s or early 1970s when a lot of them were banned. When organochlorines got banned, essentially the next group of compounds that came along were the antiesterases, things like organophosphates and carbamates. They took over. Eventually we decided that those were doing things we didn't like, and then pyrethroids took over. So these are, today they're pretty much the synthetic versions of the natural chemical. And then we'll touch on fipronil, which is one of the newer insecticides, and actually won't talk a lot about insect growth regulators, but we've moved from chemicals that have very broad activity. Organochlorines affect sodium channels in basically every organism, to compounds that are much more specific. So fipronil, and we'll talk about it, is a GABA. It's active at the GABA receptor in the nervous system. It's really specific for the insect or arthropod GABA receptor, so it doesn't have very much toxicity to mammals or other vertebrates. And then finally, the insect growth regulator. These primarily act on chitin synthesis. So when insects grow, they have to be able to make chitin to make their exoskeleton. If you block chitin synthesis, they can't molt and grow a new skeleton, and basically they'll end up dying. Well, almost no other organism makes chitin, so these are now very specific for insects and have much lower off-target toxicity than the non-specific ones like organochlorine. So we think about organochlorines, and we kind of talked about these briefly, but they were used very heavily from the 40s to the 70s. Mostly they've been phased out in developed countries, although in developing countries they're still pretty widely used, and actually just recently the World Health Organization has again begun promoting the use of DDT to control malaria in terms of being one of the more effective ways that they have it. The real problem with organochlorines, as I said, they act on all different classes of organisms. So they basically bind to the sodium channel in the nervous system. So if you look at a nerve, for a nerve to fire, it has to be able to move sodium in and out of the cell in order to change the membrane potential. The organochlorines primarily bind to the sodium channel and prevent the nerve from repolarizing or from getting sodium levels back to where they're supposed to be, and you end up in animals that are acutely poisoned with seizures because the nerves continuously fire. But they're very persistent environmentally as well. So they have very low specificity. They act on a broad group of compounds, and they're very, very stable compounds, which was originally thought to be a benefit. You know, you could spray DDT and it would be active for a long time. I live in a house that was built in 1950, and basically they poured chlordane around the foundation to keep the termites out. The chlordane is still there. The chlordane is still keeping the termites out, which is probably a good thing. It's down in the soil. We're not really exposed to it, but they are very, very stable, and they stick around in the environment for a long time. And in fact, you can probably find levels of organochlorines basically in all of our plasma. I mean, these are everywhere. So all of the organochlorines are lipophilic, so they're fat-loving. They're very easily absorbed by organisms, and they get very highly bioaccumulated, so similar to cadmium that we talked about last time. So low levels of organochlorines at the bottom of the food chain will begin to get accumulated as you move up the food chain, and you can get very high levels in your higher order predators. So DDT does cause acute toxicity by blocking sodium channels, and that's not really much of a problem. Some fish are fairly susceptible to acute toxicity, and certainly some insects, but you didn't see a lot of acute toxicity from DDT. That's really not what ended up causing DDT to get a bad reputation. It's relatively non-toxic in birds. The lethal concentration for most birds is somewhere around 800 to 2,000 milligrams per kilogram. So you need a lot of DDT to actually cause toxicity to birds. Not so with invertebrates. It was quite toxic to a lot of invertebrates. So places where you were using a lot of DDT, you might really wipe out some of your invertebrates. It usually is not going to be found in water. It is lipophilic. Almost all of it's going to be bound to sediment. And we talked about bioconcentration factor briefly in the last lecture, but essentially you can calculate bioconcentration factor, as I said, by looking at the concentration in the water, in the organism divided by the concentration in the water. And for DDT, this can range up to about 1 million. So you can have very, very high levels of bioconcentration of DDT in some organisms. And that's really what you expect to see with things that are lipophilic and stable. They tend to get bioconcentrated very well. And this would be true for almost all of the organochlorines. I'm using DDT as a poster child because we're going to go through an example with it. But it's true for dieldrin, it's true for toxaphene, it's true for all of the organochlorine pesticides. One of the things to remember about DDT is that it gets metabolized fairly rapidly. It undergoes metabolism both in the body as well as photodegradation, environmental degradation. So we have the parent compound here, para-DDT. It fairly quickly breaks down, at least in the body, to something called DDE. So you're getting basically loss of one chlorine. So you're going from this trichloro to a dichloro group here. And DDT is what you're going to find almost everywhere today. So there's not a lot of DDE. There's not a lot of DDT left if it's not being used. But sites that long ago sprayed DDT will still have fairly high levels of DDE. This can be further metabolized actually to DDD, which is illustrated here. One of the interesting things about DDD is it's actually sold as a veterinary drug for dogs with Cushing's disease, basically. So it is active on the endocrine system, which will be interesting in just a second. And I think most people have kind of heard the story about DDT. As I said, the problem really wasn't it wasn't acute toxicity. We didn't see lots of birds, lots of fish, lots of mammals dying off because they were sprayed with DDT. But what people noticed over time, and it was really prompted by Rachel Carson's Silent Spring, which is one of the first books documenting some of the environmental impacts of pesticides, was that there was a real decrease in raptors especially. The populations of these predatory birds began to drop. And it turns out that actually the breakdown product of DDT, DDE, acts as an endocrine disruptor and causes eggshell thinning. And it's thought to act by affecting the levels of parathyroid hormone, which is involved in calcium mobilization and calcium deposition, or perhaps the prostaglandin synthase enzyme, which is also involved in depositing calcium into the eggshell. And it's important for birds to have an eggshell that's the right thickness, because birds incubate their eggs, right? They sit on them. If your eggshell is not thick enough, when you sit on it, they break, and then you don't have a viable offspring. So there seemed to be a population decrease that was correlated with thin eggshells. And it looked like it was due to exposure to DDE, or accumulation of DDE due to spraying of DDT. And if you look at this particular figure down here, the number refers to the year, the wordings are places. But what you see is basically eggshell thickness. So you can see that when DDT was being heavily used in the late 1960s, eggshells were about 20% thinner than they were supposed to be in this case. And then once DDT was banned, you can see that eggshell levels gradually returned to normal, so that it took about 20 years, but basically by 1990, at least in these sites, eggshell thicknesses had actually returned to normal. And you see similar data at sites over here showing that once DDT was banned, basically DDE levels dropped and eggshell thicknesses recovered. So that was pretty well recognized in the 1960s, and as I said, things were banned in the 1970s. So why do we still want to talk about DDT? Well, Florida happens to be home to a Superfund site based largely on DDT. So in central Florida, there's a place called Lake Apopka, which happens to be about the fourth largest lake in Florida. So it's a very large lake, shaped kind of like a teardrop. There's a place down here called Gourd Neck. And actually in this area, there's a company called Tower Chemical Company that used to manufacture DDT and another pesticide called dicafol. Well, in 1980, they spilled large amounts of DDT into Lake Apopka. So they were disposing of their byproducts somewhat inappropriately, and in a rainfall event, it made its way into the lake and ended up with large amounts of DDT dumped into the lake. Subsequent studies showed that the alligator and the fish populations declined dramatically in response to this in the lake. And they showed that females actually had very high levels of estradiol in the plasma, and their ovaries were abnormal. The males had low levels of testosterone and decreased testes and penis size, so they were significantly estrogenized. Basically, the male alligators were not really male anymore. They were beginning to look more like female in some cases. And then in those alligators, 80% or more of the eggs that were laid failed to hatch. They had very high mortality rates, which was attributed to pretty much poor egg quality due to disruption of the endocrine system, not producing appropriate yolk and other factors to make that egg viable. So we had this spill that led to basically lake-wide decreases in alligator population due to endocrine effects of DDT and or DDE at this site. So this was recognized as a Superfund site. It's been pretty well cleaned up, at least at this site. And this is just an illustration from the EPA document showing basically what happened. They used to take their waste and essentially put it into a percolation pond. Unfortunately, they put that percolation pond into a sinkhole. So a lot of that material migrated directly down into the aquifer and actually contaminated some of the wells around there. They also had an area where they would burn and bury some of it, and a lot of that during heavy rainfall would make its way into a drainage, run off into a drainage ditch and make its way into the lake. So basically that Gourd Neck, the bottom end of the lake, ended up with heavy contamination from this site. Now, this is a picture of sediment concentrations in Lake Apopka from about the mid-1990s or late 1990s. And as I told you in the last graph, this is Gore Neck down here, right? This is where Tower Chemical Company was. So the darker the color, the more DDT equivalents, the total DDT. This would be a combination of DDT, DDE, DDD. There is. So the dark color is not in Tower Chemical Company. The dark color is actually all along this north shore, completely on the other side of the lake from Tower Chemical Company. So they have cleaned up that Superfund site, but it turns out that this area all along the north shore of the lake was actually farm. So in the 1930s and 40s, they used to all be lake. They drained about 14 or 15,000 acres, drained that lake, built a dam or a dike, pumped all the water out, and used the resulting soil, which was highly, highly organic muck, in what was called a muck farm. This was very rich soil, produced huge amounts of vegetables. Almost all was used as vegetable farms, and you can see that they grew corn, radishes, carrots, green beans, cabbage, all sorts of vegetables on these sites. And based on documents from extension agents and things at that area, in 1967, this is before any of the bans on DDT, you can see that a typical crop of sweet corn grown on these sites would use about half a pound of chloridane, 13.5 pounds of toxaphene, 68 pounds of DDT, and 11 pounds of parathion per acre. Right? And they would grow 2 crops of corn a year. So every acre was having somewhere around 130 pounds of DDT applied to it as normal practice. And now this was being sold in the grocery store. Everybody was eating it, right? But they had to use such high amounts because by this time DDT had been used for quite a while. Insects became resistant. You had to keep using more and more and more in order to overcome that resistance. So basically what happened on this North Shore area is that for 40 years or more they were applying very high levels of pesticides, which were really stable, right? I mean, these are stable pesticides. So they're very high levels of pesticide residues in the soils at this site. And it's somewhat of an anomaly, but actually because the areas were so large, much of it was sprayed aerially. And if you look at some sites near the airport, which is probably where they would dump the tanks when the airplanes came back, the soil had 3% toxaphene. All right, so normally you measure pesticide residues in the parts per billion range, not the parts per 100 range. So basically, 3 grams out of every 100 grams of soil at some of those sites was toxaphene. Basically, in the mid to late 1990s, Florida decided that they wanted to return that area to its natural state. So they purchased most of that land and reflooded it to convert it back to marshland. And when they did that, they were somewhat surprised that they ended up with a very large bird kill. So water birds came in, including a lot of wood storks and other endangered species, and hundreds of birds ended up dying of acute mortality at this site. And it turned out that actually it was because there was still high levels of organochlorine pesticides in the soil. When they reflooded that, basically it allowed the food chain to recover, and you again had bioaccumulation of those pesticides up to the higher order predators, which happened to be birds. So the end result of studies that were conducted here at UF showed that most of that bird die-off was actually due to acute toxicity of toxaphene, or probably mostly toxaphene, which was quite neurotoxic. So since then, they've undergone a lot of efforts to remediate this site. And it turns out that along with the acute toxicity, people have found that at this North Shore area, as well as other sites along the Ocklawaha River chain, which were similarly used for farming, there are significant problems on fish populations and fish reproduction. So when people go in and stock these areas, what you find is that the adult fish survive, but that there's little or no juvenile recruitment. So basically, new fish are not being produced each year. You have adults, but you don't have young ones. So there's very poor reproductive success. And if you look at these sites, you find that the fish have significantly altered steroid levels, and significantly altered endocrine function, very much like what we saw with the alligators in Lake Apopka subsequent to the Tower Chemical Company spill. So you see a lot of sort of chronic toxicity that actually was totally unexpected based on toxicity testing at the time. So I think it's kind of a testament, unfortunately, to how toxicology has advanced. So in the 1940s and 50s, when DDT was developed, the way toxicologists tested things was basically expose some mice to it and see if they died. And that was the extent of our toxicity testing, and we didn't necessarily evaluate other long-terms or more chronic effects of potential effects of the pesticides. And unfortunately, we're still dealing with some of that today. But you got to remember that it is a very effective pesticide. It really changed the world. I mean, we probably wouldn't have been able to build the Panama Canal without it, at least not without significant loss of life. And there's still potentially hundreds of thousands of people a year being saved from malaria by use of DDT. So as we think about pesticides, they can cause a lot of adverse effects, but they do have benefits as well. So people began to realize that the organochlorines were not particularly nice things to have in the environment, especially with the problems with the birds. So when organochlorines were banned, we had to have something to take their place. And the chemicals that took their place were largely the organophosphates. So these are all derivatives of phosphoric acid. The first one was parathione, but then other ones, things like malathione, chlorpyrifos, diazinon, all came into widespread use. So basically these were designed to replace organochlorines. The benefit they had was that they broke down relatively quickly in the environment. So the organochlorines were really stable, stuck around for years or decades. Organophosphates essentially break down in a couple of days. So the parent compound is gone. All of them act primarily by inhibiting acetylcholinesterase. So instead of acting by binding to the sodium channel, these actually block cholinergic transmission. I'll talk about that in just a minute. But another advantage of that when they were first introduced is that there was a lot of resistance and cross-resistance to the organochlorines. All of a sudden you had this new class of pesticides which overcame that resistance because it had a new target. One of the things to remember about a lot of the organophosphates is they actually have to be metabolized to an active form. So this is parathione up here. The parent parathione actually has a sulfur double bonded to the phosphorus. The sulfur, this form won't actually inhibit acetylcholinesterase. You actually have to metabolize it to replace the sulfur with an oxygen. It's called desulfuration. It's conducted primarily by P450s, and I don't know if you guys talked about P450s, but it's one of the major metabolic enzymes in the liver. And it produces what's called the oxygen analog, or the oxone. So the chemical would be paraoxone, and that's the chemical that actually binds to acetylcholinesterase and inhibits it. One of the things to remember about organophosphates is they actually will bind permanently to the acetylcholinesterase. They undergo a process called aging, and basically that enzyme is killed, and it can't recover its activity once that happens. So an organism has to produce more cholinesterase. The other group of compounds that acts in a similar way were the carbamates. So the organophosphates were derivatives of phosphoric acid. The carbamates were derivatives of carbamic acid. All of them are esters of carbamic acid in this case. They also act by blocking acetylcholinesterase. The difference is that they're reversible inhibitors. So carbamates don't bind permanently to the enzyme. They cause a temporary inhibition, and then they will break down and fall off the enzyme, and the organism will recover cholinesterase activity. And there are a wide variety of carbamates as well. Some of the more common ones would be aldicarb, carbofuran, carbaryl, which was a chemical name sold under the brand name 7, furidan, as well as a lot of others. So this is just a diagram of basically cholinergic neurotransmission. So I said that these act by blocking acetylcholinesterase. So if you look at a nerve that cholinergic nerve. It's acting by releasing acetylcholine. So acetylcholine is released at the synapse, right? So we have one nerve here, and then we have another nerve here, and we got to get the signal from here to here. So acetylcholine is released, it diffuses across the synapse, and it binds to the acetylcholine receptor on this nerve, and when it does, it propagates the action potential down that nerve. So this neurotransmitter is really important. But in order to stop that activity, you have to break down the acetylcholine. And acetylcholinesterase is the enzyme that does that. So acetylcholinesterase cleaves this into acetyl-CoA and choline, which isn't active at the receptor anymore, and it stops the nerve impulse. But if you block acetylcholinesterase, you just continually activate these receptors, and you end up with all sorts of responses. The typical acute toxicity is referred to as SLUD, salivation, lacrimation, urination, and defecation, because it activates all of your different parts of your primarily parasympathetic nervous system. So your foam, salivating, your tears are running out of your eyes, you can't control your urine output or defecation. You have real bad diarrhea and things. And this is just a diagram of acetylcholinesterase, essentially showing that it has two sites, an anionic site and an esteratic site. This is acetylcholine. It gets cleaved into acetic acid and choline. But the organophosphates and carbamates are almost perfect mimics for this and fit right into the site very, very well. And we'll block it, either reversibly or irreversibly. So when they do, they allow the buildup leading to the toxic symptoms. And there are a number of things to remember about sort of susceptibility to acetylcholinesterase inhibition when we're thinking about toxicity. One is that there's a lot of organism-to-organism differences in susceptibility, but there can also be differences in stage-specific susceptibility. As I mentioned, they have to be able to produce this oxygen analog, the oxone, to actually be active. So if we look at sort of toxicity of this in midge larvae, you can see that chlorpyrifos, and this is chlorpyrifos oxone, that the second instar is different than the third instar, which is different than the fourth instar. So these are different ages of larvae, and there's big differences in terms of susceptibility. Whereas we call the LC50, right, the one that causes 50% toxicity, for fourth instar larvae would be very, very susceptible, somewhere less than a nanomolar. Whereas ones that are only second instar would be almost 100 nanomolar, so almost 1,000 times difference in terms of susceptibility between different ages of organism. Now, another group that came along was the pyrethroids. These were derivatives of the natural pyrethrin that came from, as I said, from certain types of chrysanthemum. These were back to being sodium channel agonists, very much like the organochlorines, but these were designed specifically to act on arthropod or insect sodium channels. So they have very low toxicity to mammals. Like the organophosphates, they have low environmental persistence. Basically, UV radiation breaks these down very quickly. A lot of times they are actually combined with a compound called piperonyl butoxide, which is a cytochrome P450 inhibitor, to promote, to extend their ability to act. So it increases their action. And there's a lot of different types of pyrethroids. Permethrin is probably one of the most widely used, but bifenthrin, deltamethrin, resmethrin, anything with thrin on the end of it would be probably a pyrethroid. And these have essentially taken over almost all the market from organophosphates and carbamates. So we banned organochlorines in the 1970s. In the 1990s and early 2000s, we pretty much got rid of diazinon and chlorpyrifos, which were the major organophosphates that were widely used by homeowners for insect control, both on lawns and spraying Raid and things like that in your house. EPA banned those, and pyrethroids have pretty much filled that market niche. So most of what you use in your house for insect control today is going to be a pyrethroid. And that's where I want to kind of start with one of our case studies, talking about looking essentially at sources of pesticides in aquatic systems. And basically, we know that pyrethroids have become very common with homeowners. And we know that you get landscape irrigation and stormwater runoff can actually carry significant amounts of materials that we use as homeowners into the aquatic environment. So people began asking the question, does residential use of pyrethroids result in essentially sediment concentrations that could cause mortality in sediment toxicity tests? And the way they looked at this was to take Hyalella azteca, which is a very commonly used test organism. It's an amphipod that lives in sediment. It'll run around on top of the sediment. Sometimes it'll burrow a little ways into the sediment. And they expose them to the sediment, different sediments, for 10 days, and essentially look at how many of them are left at the end of that 10-day test. This particular study took place in Placer County, California, in 2003. So they were looking at pyrethroid use. And the thing to remember when you look at use, it's really only for commercial use. People don't ever record the amount of pesticide used by homeowners. So almost all the reporting is done by commercial use. But you can see that there were large amounts of pyrethroids used in Placer County, even for commercial applications. So this was kilograms per year. And you see that cypermethrin for structural pest control, basically termite treatment, was the biggest use, followed by permethrin. And then permethrin was very widely used on lawns, basically, or flowers for landscape maintenance. So basically what they did is take sediments from a variety of different sites around the county and compared what they found in agricultural areas to what they found in more residential areas. And essentially this is just a map. So if you look, you can see that a lot of these that have what look like subdivisions around them would be residential, and these areas out here that are more agricultural areas. So they took that sediment, exposed Hyalella to them. Now, so they're actually taking field sediments, and then they went back and they looked at what happened to those Hyalella. And what you see is that all of these pink dots were sites where over 70% of those sediment organisms died. So this would be sediment that was acutely lethal to insects. And what you find is that in most cases, those are actually associated not with agricultural areas, but with highly urbanized areas. So basically they were able to show that there was significant runoff of, and they were able to attribute it, in this case, to the pyrethroids by measuring the pyrethroid concentrations in the sediment. Significant mobilization of pyrethroids from homeowner use, both around the home and on landscape. If you go out and look at those field sites, you find that there are very few crustaceans or amphipods present. So, homeowner use of pyrethroids can really have devastating impact on those communities. And the outcome of this was that the majority of the input in this case was actually from residential application rather than from agricultural runoff. And a lot of this is because they're fairly, well, if people abide by the rules, there are fairly strict controls for agricultural use as well as engineering controls to minimize runoff, whereas many times homeowners don't have the same engineering control or restrictions on the use. And a lot of us suffer from the idea that the little bit is good, more is better. So we'll put a lot more on than we're supposed to. All right. Now I want to touch briefly on fipronil. So fipronil, as I said, is one of the newer compounds even after pyrethroids. And this is now probably one of the most widely used pesticides for termite control. You've probably heard ads for Termidor. On the radio, Termidor is basically fipronil. Fipronil is used for flea control on insects, on pets, I'm sorry. It's not really made its way into other homeowner products yet, primarily pest control, but it's also fairly widely used in agriculture. Really wasn't introduced widely until the 1990s, and this one acts in a different way. It's actually a GABA antagonist. So GABA is gamma aminobutyric acid. It's another neurotransmitter. Tends to be an inhibitory neurotransmitter. So when a nerve releases GABA, it prevents another nerve from firing, so it provides inhibitory input. And if you block the action of GABA, you get what's referred to as disinhibition. Basically, you're inhibiting the inhibitory action, so you get excitation as a result of that. And this one is very, very toxic to non-target arthropods. It's very specific for the insect GABA receptor, so it has very low toxicity in vertebrates, which is one of the reasons why you can basically dump it right onto Fido to prevent fleas, because Fido is not going to respond to fipronil, but the flea will. But it is really toxic to other aquatic organisms, especially fish and shrimp. Shrimp and crayfish and things, much more so even than fish. It does have some effects beyond GABA, at least beyond acute toxicity. They may be actually mediated by GABA antagonism, but there's been evidence that it can cause some reproductive and neurodevelopmental abnormalities in aquatic organisms. And one of the things to remember about fipronil is that it's actually what's called racemic. What you buy is a mixture of enantiomers. So enantiomers are different chemical forms. They're exactly the same chemical, but they have different optical properties. So you have an R and a S, or a plus and a minus enantiomer. And fipronil is a mixture of that, but the enantiomers actually have quite different toxicity. So if you look at what we see with fipronil, just kind of illustrate some of these points. We can see here is the concentration in micromolar of fipronil. Once we reach about 230 nanomolar, we start to see significant decreases in growth in fish larvae that are exposed to fipronil. And this happens at a much lower concentration than where you start to see some of the more overtly neurotoxic responses, like effects on swimming activity. This is just illustrating the different effects of the enantiomers. So you can see there's a plus enantiomer, a minus enantiomer, and the racemate basically is a mixture of the two. It's what you would buy typically. And you can see here that the plus enantiomer is actually considerably more toxic than the minus enantiomer. And almost all of the toxicity of the racemate is much more similar to the minus enantiomer. And this is just basically evidence that it's actually binding to the GABA receptor. BIDN is another GABA receptor blocker. If you add that to it, you get an increase in toxicity, but it's showing that it's not binding to the same site on the GABA receptor. Now, one of the important things about fipronil to remember is metabolism, but not necessarily metabolism in the body. One of the major pathways of fipronil degradation is actually photochemical degradation. So when it's exposed to sunlight, it breaks down. This is the fipronil parent. This is what you will get. The sulfone is what you'll get in the body. So fish, insects, us are making the sulfone. But UV light can make a variety of other products, including the sulfide and this compound called disulfonyl fipronil. And it turns out that disulfonyl fipronil is actually much longer lived and much more toxic than the parent compound. So very much like what we saw with DDT, you have to worry many times about metabolites or breakdown products when you're thinking about toxicity of pesticides. It's not always the parent compound that causes the problem. So if we look at an example of where fipronil toxicity occurs, sort of aquatic toxicity of fipronil, we take a look at what's called the Mermentau River Basin. So this is in southern Louisiana. Basically, there are very large parts of the bayous here that are grown for or cultivated for rice. Some 400,000 acres in that basin. And when you grow rice, basically you alternately flood and don't flood the field. And people there figured out that they could double crop that land with crayfish. So crayfish is a huge export, and actually for many of these farmers, crayfish is the major cash crop, not rice. But basically what they would do is when they flooded the fields to grow rice, they would put in a bunch of crayfish. The crayfish would grow, and then when they drained the field to harvest the rice, the crayfish would burrow into the ground. Some of them would sort of overwinter and come back out the next year. Other times they would stock them with additional crayfish. And it was an important part of the income for those people, especially when they had droughts and things that really had an adverse effect on rice farming. So everything was going along very well, and then sometime in the 1990s, they started to notice that they had a lot lower harvest of crayfish from some of these fields. In fact, they started to find a lot of dead crayfish turning up, and people were looking all over for the source of this crayfish mortality. It was getting worse over time, and they were beginning to wonder if it was a new disease or some other thing that was causing the problem. And eventually they started to look at fipronil, which was the new pesticide that had been introduced into rice. So what they began to do with fipronil was actually treat the rice seeds with fipronil to prevent insect damage on the seed before it was planted and while it was first planted. So they treated it with fipronil. The brand name of that product was Icon. And they found that, not surprisingly, that rice that was treated with Icon had high levels of fipronil and fipronil breakdown products, but you didn't see these in the untreated seed. So this is just data showing the concentration of fipronil in treated and untreated seed, and you can see that in the seed anyway, most of it is present as the parent, but there is already some of the breakdown products, the sulfone and the sulfonyl fipronil. Now, once you plant that rice seed that's been treated, what you would expect to see happen is that fipronil would, basically come off of the seed and make its way into the water, and that is indeed what you find. And when they looked at the water concentrations of fipronil and the breakdown products in fields that were planted with seeds that had been treated with Icon versus ones that were not treated, you saw significantly higher levels of fipronil as well as the sulfone, and very high levels of this breakdown product, the sulfonyl fipronil. And the crayfish survival in those particular fields was only 40%, whereas in the fields that were planted with untreated rice seed, it was almost 85%. So they reduced, just the use of the Icon treated seeds reduced crayfish survival by 50%. Now, when you look at what was actually causing the toxicity, it turns out that most of the toxicity was actually due to the breakdown product, not the parent. So the disulfonyl fipronil, which was the photo degradation product, is the one that accounts for much of the actual toxicity you see, as well as the sulfone, which is another product that could be produced either by biologic metabolism or photo degradation. Now, if you look at this site, this was 24 hours after the seeds were planted. You see these are hazard quotients. It's basically an index of how toxic it is. If you filter that water with a 0.45 micron filter, you remove a substantial amount of the toxicity in it, so that the upper end, it drops by a factor of close to 4. In some cases 6, so you remove 80% or more of the toxicity in that case, which indicates that this is largely binding to particles. You can remove it with a filter, and this is consistent with what we know about fipronil. Fipronil has a very high, it's hydrophobic or lipophilic, very much like DDT. You measure that oftentimes with something called the log KOW, which refers to the amount, the OW is octanol water partition coefficient. So if you put some chemical into a bottle containing octanol and water and you shake it up and you measure how much of it's in the octanol versus how much of it's in the water, it gives you this measure of hydrophobicity. It's called log KOW. Well, fipronil has a high KOW, which indicates it doesn't like to be in water, and it's going to bind to sediment, which is exactly what you see here. Now, the thing about southern Louisiana and rice fields in particular is that they are very turbid. There's a lot of sediment, and that sediment can move a long way. And it turned out that that is what they found, that not only was it affecting fields that were actually planted with fipronil-treated seeds, but even at sites several miles downstream, they had concentrations above 4 parts per billion, which was enough to cause toxicity. So another thing to remember about pesticides is it can bind to sediment, it can be transported a long way, and that the breakdown products can also cause toxicity. And in this particular case, the breakdown product is longer lived and probably more toxic than the parent compound to the aquatic crustaceans. All right, now the last one I want to talk about is probably one of the most controversial chemicals on the market today, is atrazine. And atrazine is an herbicide. It's the most widely used herbicide in this country anyway in terms of volume of use. It's produced by Syngenta, so we'll talk about who's funding studies as we go on, and you'll hear Syngenta again. But Syngenta is a large agricultural chemical company, and it's a very important economic product. I mean, the market for atrazine is well over $400 million a year. In the United States, about 70% of the corn that's grown here is sprayed with atrazine. So that's some 62 million acres is actually sprayed with atrazine. A lot of that atrazine runs off of the field and makes its way into waterways, and you can see it all up and down the Mississippi River Valley. And not only there, but you can find measurable amounts of atrazine in almost every surface water in the United States. So if you go out and measure a creek, basically anywhere in the United States, you'll find atrazine in almost all of them. Typically the levels are very low though, and usually well below the 3 part per billion level that's been established by EPA. As an herbicide, it tends to not be very toxic to animals. Most herbicides act on processes that are more specific to plants. So it's nearly non-toxic to birds, fish, and mammals. But there's a raging debate going on about its effects, its long-term effects on frogs, and also potential effects on fish and mammals. And we'll talk a little bit specifically about some of the frog data. So this is just a diagram of the Mississippi River Valley, which is where a lot of the corn crop is grown in the United States. And basically what you're looking at here is a calendar year: February, April, June, August, October, December, and then atrazine levels measured in different levels of the Mississippi River. And what you see is that in the spring, when we plant the corn crop, basically levels of atrazine spike, and then by the time we're not so worried about applying herbicide anymore, the levels of atrazine go away. So you have significant runoff of atrazine into waterways. And this has sparked some interest in whether or not atrazine may be responsible for some of the declines that are observed in amphibian populations. You've probably heard various places that amphibian populations are in decline pretty much around the world. Frogs as well as salamanders and a lot of other amphibians are in pretty bad shape. So people started looking at whether atrazine might have any effect that could be linked to this, because there was some evidence that atrazine might actually have endocrine-like effects in some of the studies that were conducted with atrazine. So if you take a look at, this is a control testis from a frog. You see basically tissue that's producing sperm here. If you look at a control ovary, you see lots of developing oocytes, right? Like they're supposed to be. The females are making eggs, the males are making sperm. If you then treat these frogs, at least in this report, with 0.1 parts per billion of atrazine. Now this is well below the 3 part per billion level that EPA established. This is a picture of the testis. If you then look at that histologically, you don't see all spermatogenic tissue. You actually begin to see oocytes in the testis. So this is a condition referred to as ovotestis or testis ova, or intersex, where you have gonads that aren't doing exactly what they're supposed to be doing. And this type of data would suggest that atrazine, very, very low concentrations of atrazine in this case, are able to badly disrupt the endocrine function of these frogs, to the point where the gonads not only are not producing steroids, but they're actually beginning to be mixed up between testis and ovary. Now, one of the things that's difficult to deal with in terms of some of the atrazine literature is that you don't get a normal dose response. So I think you've talked about dose response, and typically the idea is that the more you get, the more of a dose you give, the higher your response is, right? So what you see in this graph is just two levels of a dose response curve, but the black parts of the bar indicate gonadal dysgenesis, basically any of the gonads, either testis or ovary, being messed up. And the blue bars are specifically testes that actually have oocytes growing in them. So in control animals, there's a very low level of gonadal dysgenesis and no evidence of testicular problems. With a very low concentration of atrazine, 0.1 parts per billion, we see that up to nearly 70% of the males have some type of gonadal problem, with about half of that having problems that are testis with ovaries or ovarian follicles growing in them. However, if you increase the concentration 250-fold, that level drops dramatically. So we're not seeing a standard dose response in this case, right? We go from a low dose to a high dose, and the response is not going up. The response is actually going down. That's problematic from a toxicological point of view. And one of the things that people have invoked to explain responses like this, and I think you've already touched on it, is what's called hormesis, or the inverted U-shaped dose response curve, inverted dose response curve. And what you get here is a situation where the red bar is indicative of normal, and at very low doses you can actually get a change in function that then returns to normal, and at higher doses again departs from normal. So hormesis is clearly a very real response, at least in some areas. If you look at radiation exposure, for example, you've probably heard that some people will go down into mines to get low doses of radiation. It acts as an immune stimulant, and there's pretty good evidence that very low doses of radiation can actually cause increased function of immune cells, at least in vitro. I'm not going to say yes or no in terms of whether I believe it works in vivo. But then, of course, very high doses of radiation will cause acute radiation poisoning, right? Toxicity. You see the same thing in a lot of organisms during reproduction, where low level of stress will actually cause increased amounts of reproduction, but a much higher level of a stressor will then shut it down. And I don't think that's terribly surprising, given that an organism is sensing stress, and perhaps in an increased effort to transmit its genes and produce offspring, it puts more emphasis into reproduction. But this response has been termed hormesis, and it's possible that this could be active with atrazine. This is another piece of data from some of those studies, and what you see here is a map of atrazine use across the United States. The more yellow to red it becomes, the higher the levels of atrazine that are used in those counties. And of course you can see that the darkest red is in what we would call the Corn Belt. This group actually took samples of frogs from various places across the United States, from places that had essentially no atrazine use or very low atrazine use to areas that had very high atrazine use. And then they measured contaminant level, atrazine itself, as well as some of the breakdown products. And then they looked at gonadal problems So these were field samples that were correlates to the lab study that we talked about a second ago. And again, what you see is that they found sites with very high levels of testicular oogenesis, but those sites didn't seem to have a direct correlation with contaminant level. Right? So sites 2 and 3 had approximately equivalent amounts of atrazine, but site 2 actually had maybe 10%, whereas site 3 had about 90% incidence of testicular oogenesis. And the sites with the highest levels, much higher levels, actually had very low levels. So this leads to real difficulties in interpreting this data, I think, in terms of, is atrazine causing this testicular oogenesis you see in these frogs? Certainly it's being seen in field sites, it's being seen in laboratories. It doesn't necessarily seem to be dose responsive in either field, or typical dose responses in either field or laboratory settings. This has led to a lot of debate because there's concern also raised about its potential impact on mammals as well, including humans. So this debate has raged on for nearly 10, well, probably over 10 years now. But one of the big problems is this lack of a typical dose response. It's very difficult to interpret when you can't see a standard dose response. A lot of this work was actually done by one scientist, Dr. Hayes. Other scientists have not been able to replicate a lot of these results. That's typically not a good thing scientifically, but this issue is compounded because a lot of the other scientists were actually funded by Syngenta, who is the maker of the chemical, which then raises questions about conflict of interest. So it's very difficult to interpret. EPA has took a good hard look at it, and in 2007 concluded that atrazine was not responsible for affecting amphibian gonadal development based on the results of 19 compiled studies. At that point, I pretty much thought atrazine had been laid to rest, but it's now back. EPA's Science Advisory Council is meeting about atrazine about 8 times this year. So atrazine is still, the debate around atrazine is still raging. And in Europe, it's actually been essentially restricted or nearly banned since 1998. The Europeans tend to take a very different view of toxicology than the United States does. For better or for worse, they use what's called the precautionary principle, where you say if there's evidence that it may cause a problem, you try to prevent it, as opposed to having to provide evidence that it actually does cause a problem. So just to wrap up, pesticides, I think the things I'd like people to remember are that it's not only pesticide parent compounds that cause toxicity, it's very often the metabolites. So of the ones we talked about, the organochlorines, a lot of the chronic toxicity is due to breakdown products like DDE. With the organophosphates, it's actually the oxygen analog that causes most of the acute toxicity. With fipronil, it's actually the breakdown product. With atrazine, we have no idea. But remember that these things can move a long way. A lot of them are hydrophobic. They bind to sediments. And as that sediment moves, so does the chemical move. And it can then be accumulated through the food chain because many of them are quite hydrophobic and actually do get bioaccumulated. Or their metabolites can be bioaccumulated. A lot of what you see in terms of toxicity with pesticides is aquatic invertebrates because they share so many similarities with the actual target organisms. So you see these non-target species that are still arthropods actually being really susceptible, probably the most susceptible population to a lot of pesticide toxicity. I think it's pretty clear that non-standard toxicological findings cause a lot of debate. So things like atrazine, where we get responses that we don't understand, we really don't know what to deal with. And then this raises, I think, a couple of questions, and we won't answer them today, but things just to think about is we get down to the question of what's really ecologically significant. So if you look at the way that we regulate things, basically we say that the solution to pollution is dilution, right? So that you're allowed to have a certain amount of a chemical. We say below that amount, it doesn't matter. Above that amount, it's a problem. As long as you stay below that amount, it's okay. And typically what we look at when we're determining what that level should be, we look at fish populations, invertebrate populations, sometimes earthworms, but we still don't have a real good handle on doing real long-term studies. It's hard for us to do those just because of time and expense. And it gets to the question of what is ecologically significant. I mean, there's a lot of debate going on now about sort of ecosystem processes and things like that. We don't have a good way of addressing all of that. So we typically regulate based on, does an organism live or die? Does it reproduce normally? So, is that appropriate? And I think then it gets to the questions that we see with some of the frogs and some other questions. If we know that it can cause an effect, but it doesn't affect the population level, so it's producing effects in an individual, but it's not affecting the population, is it a problem? I think that's the flip side of that question. So when you look at human health, we worry about the individual, right? If one person gets cancer, we're concerned. When we look at environmental or ecological health, we don't care about the individual, we care about the population. So if you have things that they produce effects, but they don't affect population stability, is that a problem? Answer. Nobody, I think, knows the answer, but something to think about anyway.